Good morning, everyone. We're going to get started. Thank you. Bonjour tout le monde et bienvenue à la locution d'ouverture pour la séance en sciences fondamentales de la part de ma coprésidente, Dr. Christina Guzzo. Je me présente, Dr. Jimmy Dikiakos de Western, et je vous souhaite la bienvenue. Welcome, everyone, to the opening address, the keynote talk for the Fundamental Sciences, Basic Sciences session at CAHR 2024. It is my absolute honor to introduce Dr. Paul Bieniasz. Paul Bieniasz is the Pernell W. Chopin Professor and Head of the Laboratory of Retrovirology at the Rockefeller University and an HHMI investigator. He received a PhD from Imperial College, University of London, and was a PDF Fellow at Duke University. He was previously a faculty member at the Aaron Diamond AIDS Research Center. His laboratory has contributed in multiple areas of retrovirology, including the discovery of mechanisms and proteins employed by viruses during particle genesis and by hosts as intrinsic antiviral defenses. His group has also developed novel methods to study extinct viruses, HIV-1 AIDS pathogenesis and therapy, and antibody-based immunity to SARS-CoV-2. Without further ado, let us all greet Dr. Paul Bieniasz.
Well, thank you very much, Jimmy, for that introduction. Thank you, and Christine, for the invitation to speak here. It's an honor and pleasure to talk to you this morning. Here's the boring slide. Not much to speak of here in terms of conflicts. Gosh, that's a surprise. So what I'm going to talk to you about today is how hosts defend themselves against viruses. We'll do a deep dive into some mechanisms of just a couple of examples, and then talk a little bit more broadly about how knowledge of host defense might be applied. So viruses are rarely beneficial and often deleterious to their hosts, and so evolution has endowed us with various mechanisms to fight off viruses. You will be aware of the adaptive immune system, whose key feature is that it evolves by Darwinian selection during the lifetime of an individual host. However, the mechanisms we're going to talk about today are encoded in the germline. They're invariant within an individual. They do evolve and adapt, but they do so over evolutionary time. And because that evolutionary rate is so much less than that of viruses, they, by their very nature, have to target things about viruses that are difficult for viruses to change. And that's a key feature that we can use and learn from. So, as I said, a couple of in-depth examples. Mechanistic, we're going to really get into the weeds here. I understand this is maybe a diverse audience. Some of you, I'm sorry, you may get lost, but there are other people who are quite sophisticated virologists, so I have to speak to them too. But this story begins with the study of an HIV accessory protein, VPU, very small protein and gene, something of a scientific backwater in times past because it's actually not conserved in many simian immunodeficiency viruses and actually not required for HIV replication in many situations. It's a small transmembrane protein. But what was interesting about it is that occasionally you could find cells that are designated red in this color scheme, where a wild-type virus would replicate fine, but a VPU-deleted virus would replicate fine up to, but not including, the point at which you expected the infected cell to release virus particles. Those virus particles would become trapped on the cell surface. My group found that those viruses were being held by a protein tether. We also found that that phenotype, that green permissive to red non-permissive switch, could be induced by type 1 interferon, and that led us on a hunt for genes that might be responsible. We identified them simply by comparing genes that were expressed in this type of cell but not this, and were induced by interferon alpha. And what we discovered was this protein. It's called tetherin, for reasons that will become if they're not already obvious. It's a small rod-shaped protein that's very unusual, unique in nature, in fact, in having both a transmembrane domain and a lipid glycophosphatidylainositol C-terminal anchor. So two different ways of anchoring in the membrane. Here's the clear phenotype. You take cells that don't normally express tetherin, a wild type and a VPU-deleted strain in a single cycle of replication yield equal amounts of virus. But you add that one gene to this cell, and now suddenly VPU becomes required with about a 100-fold defect. Same experiment, different cell line, but you recapitulate that tethering phenotype by expressing this single protein. What's interesting about this single protein that induces HIV tethering is it doesn't only work on HIV, it can work on essentially, in principle, any virus. So here's an experiment where we've made virus-like particles by expressing an Ebola virus protein. You express that protein in cells, but if you co-express tetherin, then you don't get particles in the supernatant, in contrast to when tetherin is absent. And then if you add VPU, you can rescue the ability of that viral protein to generate extracellular virus particles. The key point being there is zero sequence and zero structural homology between the protein components of Ebola virus and HIV. Clearly, tetherin is targeting something that doesn't require any particular sequence in viral proteins. So here's another set of experiments. So here we have a murine leukemia virus that expresses GFP growing in cells that don't have tetherin or do have tetherin. And you can see the virus spreads to neighboring cells without difficulty. Those virions trapped on the surface, fully infectious, can infect neighboring cells. But if you sample the cell culture supernatant, there's a massive deficit in the amount of free virions that could go and disseminate. And if you make a transgenic mouse that is expressing tetherin in the target cells that this virus infects, you can prolong the life of those mice that would otherwise die of cancer quite significantly. So it's a real antiviral gene that has a very particular mechanism of action. So how does it work? So it's actually turned out to be a rather difficult nut to crack, but ultimately was quite rewarding. And the key two experiments are shown on the next two slides. The first one shows that you can change the protein sequence of tetherin entirely, and it still works. So what we've done here is to take protein domains from proteins, again, that have no sequence homology to tetherin, but share structure, stitch them together in a completely artificial way to recapitulate this membrane topology, and what you get is an artificial tetherin that works nearly as well as the natural protein. So no sequence requirements in viral proteins, no sequence requirements essentially in no specific sequence requirements in the tethering protein. That enabled us to do this experiment, which I think really nails it. It's a somewhat underappreciated experiment, I think. So what we've done here is to take cells, infect them with a virus, an HIV strain that lacks VPU. The viral proteins accumulate in cells over time, but don't appear very much in supernatant unless you do this trick. So what we've done is taken advantage of the fact that you can manipulate tetherin sequence and still have it work to program it with a specific protease cleavage site, this protease factor 10A. And if you add that protease to these infected cells, you can release the virus particles from the cell surface, sort of convicting tetherin as the direct component of the tether. Just as importantly, if you put epitope tags on this tetherin protein, then they appear in the released virions. And depending on where you position them in the tetherin protein, you can deduce that this cartoon with the lipid anchor, the GPI anchor, embedded in the virion membrane and the transmembrane domain embedded in the cell is, for the most part, how tetherin traps virions on the cell surface. Okay, so that basically is the story of how we understood the HIV-1 VPU protein to enhance virus replication in particular cell types and the mechanism of action of the protein it antagonizes. It is one of many similar stories. I have to say that scientists working on HIV have really led the way in uncovering dozens, and there are probably hundreds of genes that have some kind of antiviral activity. They are weird and wonderful in their mechanisms. Every new one is a story of scientific discovery that's interesting and important. Here are just a few examples in the field of HIV. Tetherin I've just told you about. In some cases, it's not the VPU protein that is responsible for antagonizing it. So I told you that this only works in the absence of VPU. But what VPU does is actually bind to tetherin and remove it from the cell surface. In other simian immunodeficiency viruses, sometimes NEF takes on that role. Other antiviral proteins that HIV has to deal with include ApoBec3. It's a family of proteins that are cytidine deaminases. They cause hypermutation of the viral genome to an extent that's lethal to the virus. HIV and SIVs have a protein called VIF. Its only job is to bind to ApoBec proteins and to destroy them, basically remove this host defense. SAMHD1, another protein, what it does is deplete dNTPs, the building blocks required for reverse transcription. Some SIVs have a protein whose probably only job is to remove SAMHD1 from cells to allow reverse transcription. There are various other proteins at various points in gestation in understanding of exactly how they work, and I won't go into them. But many of them are targeted by HIV accessory proteins. A key role for HIV accessory proteins is basically to denude host cells of their defenses against HIV and SIV. There's another class of proteins that do have some specificity, and they invariably target the HIV capsid or mechanisms involving the HIV capsid. And that's very likely because the capsid is a structurally very compact key protein, and it's really quite difficult for the virus to evolve changes in capsid without incurring a large fitness deficit. And so there are proteins such as TRIM5 that greet the incoming viral capsid and prevent cells from becoming infected. Now, that paradigm works in a species-dependent manner. So, for example, if you take the human TRIM5 protein, it's not active against HIV-1, but it's active against certain other retroviruses. If you take the TRIM5 protein from rhesus macaques, for example, it's very active against HIV-1. And understanding these species-dependent differences brings me to the first sort of application of this type of knowledge, this understanding of how these host defenses can specialize in a host-dependent way to provide defenses against a particular range of viruses, and then the viruses try to adapt to evolve their way around those. But these types of evolutionary arms races create viruses that are specialized for particular hosts. So HIV-1, for example, as I've just said, has evaded the TRIM5 proteins in humans, but not in monkeys. Similarly, the VIF proteins of HIV-1 can destroy the ApoBec proteins that it finds in human cells, but it's actually completely ineffective against the same proteins in other species, including monkeys. We thought we could take advantage of that knowledge to actually engineer new host tropism into HIV-1. You can understand how that might be useful. We have simian immunodeficiency virus models that we can infect macaques with, but it's really a virus that's about 50% different at the sequence level to HIV-1. We wanted to try to engineer HIV-1 to infect monkeys so we can study the human virus in an animal host. And so here are a couple of attempts to do so. So here we've taken HIV-1, and what we've done is to replace its capsid protein and its VIF protein with those from SIV-MAC. So HIV-1 grows just fine in human cells, dead as a doornail in rhesus macaque cells. SIV-MAC, simian immunodeficiency virus from macaques, replicates in both human and macaque cells. But if we take this virus, which we call simian-tropic HIV-1, make these two changes, what we get is a virus that can now replicate rather nicely in macaque cells. We found a macaque species, fortuitously, pigtail macaques, that actually have a weird TRIM5 protein that doesn't inhibit HIV-1. And so to make an HIV-1 strain that can replicate in that macaque species, all we needed to do was change the VIF protein. Okay? And so now we have an HIV-1 strain, a simian-tropic HIV-1 strain, that can antagonize the macaque apobec proteins, and that virus now grows very nicely in these pigtail macaque cells, unlike the unmanipulated HIV-1 that replicates rather poorly. So this was the state of knowledge that we had about antiviral proteins at the beginning of the series of experiments that I'm about to describe to you. Key point that will become of interest later is we didn't yet know about all the work with all the interactions of VPU and tethering. That'll follow in a few minutes. So what we did was to take this virus, okay? We gave it an R5 tropic HIV envelope, actually a cocktail, but only one mattered. We made an HIV-1 strain that has SIV MAC VIF, and then when we infected pigtail macaques. And so, as I've told you, that gets around two key blocks to HIV replication in macaque cells. So we infected a couple of macaques. The virus took. It was kind of wimpy. We passed it to another macaque. And then for the next few years, we passed it in macaques. And at each stage of the passage, what we would do was transiently and partially deplete CD8 cells from the macaque along with the administration of virus. What that does is it gives the virus a little bit of breathing room to replicate in an immunologically attenuated, temporarily immunologically attenuated host, which facilitated the adaptation process. So then over the approximately the next 2 to 3 years, we passaged this virus in macaques. It gradually got hotter and hotter, and then on passage 4, one of 3 animals that was infected succumbed to AIDS. It had 2 independent AIDS-defining diagnoses. One was a B-cell tumor that was widely disseminated. It's shown here in kidney. And then in its lungs, you could see these black spots, which are pneumocystis organisms. We could take virus from that animal, recapitulate that phenotype. And so we passed that virus, and in black on macaques where we gave the anti-CD8 antibody along with the virus, they universally and rapidly progressed to AIDS. The animals that received the virus but did not receive the CD8 antibody, they became infected but went to a sort of a long-term elite controller type of status. You can see that here, plasma virus load in CD8 depleted macaques and in non-CD8 depleted macaques. Interestingly enough, if you do the CD8 depletion at a later time point, so in fact these animals have all had the same treatment, it's just that in this case the 2 treatments were divided in time, you essentially can recapitulate the extremities of clinical outcome that's observed in HIV-1 infection in humans. Rapid progression in the one case, elite control in the others. And this is an absolutely reproducible phenotype. And it implies that at least in this model, and perhaps more generally, events that are deterministic of clinical outcome can occur in this very short period of time after administration of the virus. Does the virus or the immune system get on top, as it were, and to define the subsequent clinical course? Because we can manipulate it to, as I say, arrive at those 2 extremities. So something rather interesting happened during this passage process. I said I'd come back to VPU. What I didn't tell you was that HIV-1 VPU, while it binds to and is very good at removing human tetherin from cell surfaces, it's completely ineffective against macaque tetherin. So in those early stages of adaptation, the HIV-1 VPU protein in the virus that we gave to those monkeys wasn't really doing anything. But what happened over time, and we got this from the sequence of the virus that came out in passage 4, is that VPU underwent a gain of function. It acquired amino acid shifts and substitutions in its transmembrane domain that allowed it to bind to the macaque tethering protein. And as you can see here, while the wild-type VPU basically has the same phenotype as no VPU in that if you add more tethering, you get less virus, right, for the wild-type and no VPU. But for this adapted VPU protein, pigtail macaque tethering becomes much less effective at inhibiting that virus because the VPU, the adapted VPU, is able to antagonize the monkey tethering. And interestingly enough, what we did during this adaptation process is effectively recapitulate something that has happened naturally. So I mentioned previously that many SIVs do not have a VPU protein. They instead use their NEF protein to antagonize tethering. And if you look in the simian immunodeficiency virus world, you will find SIVs that don't have a VPU and use NEF as their tethering antagonist, and you will find other SIVs that do have a VPU and they use it to antagonize tethering. Now, it turns out that SIVs from both of these groups are the ancestors of SIV-CPZ, the chimpanzee virus, which is in turn the ancestor of HIV-1 in humans. And in SIV-CPZ, it's in fact the NEF protein that takes on the role of tethering antagonist by binding to this peptide motif. This peptide motif actually happens to be missing in human tethering. So when SIV-CPZ arrived in human populations 120 or so years ago, it had to reacquire this activity of tethering antagonism, which it has done so in very much the same way as happened in our macaque passage experiment. So what happens in adaptation in the laboratory can often recapitulate what happens in nature. Nature does more gain of function experiments every day than humans can conceive in a lifetime. Okay, but now knowing this and having our adapted virus, to have a virus that we can actually use as a model system, what we need to do is to make a clone of it so we can make virus stocks and infect monkeys with a homogeneous viral stock. And we've done exactly that. We took virus from the animal, the first animal to die from HIV-1, and made a molecular clone, and it recapitulates that phenotype. So if we give CD8 antibodies and virus, monkeys inevitably rapidly progress, losing all their CD4 cells over a matter of months. So what can we do with such a model? So this is one of the things we've done, and this is in collaboration with scientists at Gilead. And I should point out that all of our work in monkeys, the simian immunodeficiency virus work, is done in collaboration with Jeff Lifson, Greg Delpret, and their team members at NCI Frederick. It's been a long-standing and very fruitful collaboration, which most recently has culminated in this experiment. This is a rather new medicine, lenacapavir, that works by targeting the capsid protein of HIV-1. And in so doing, it blocks the import of HIV-1 into the nucleus. It's a rather remarkable molecule because of its potency and the fact that you can formulate it to be long-acting. And so this is a PK experiment done in pigtail macaques. Okay, so they've been given a couple of doses of lenacapavir, and then the drug is maintained at concentrations that are expected to be protective for months, more than 6 months after administration of the drug. So does it work to block HIV-1 infection? Yes, it does. It does very effectively. So here are pigtail macaques that have been infected with that cloned simian tropic HIV-1 strain that we've derived. They all become infected uniformly. Regular prophylaxis, PrEP, works well in these macaques. This is an equivalent of a once-daily tablet that these macaques take. And then here's lenacapavir, given a single injection, and then a month later, the monkeys are challenged with 1 million infectious units intravenously, probably the highest dose challenge one could possibly conceive. yet they may remain completely protected. There's no sign of viral nucleic acid in these macaques at all. So when you consider that this medicine could potentially provide this level of protection for months after a single injection, recognizing that performance is actually substantially better than many viral vaccines that we take, you come to realize that this type of approach really could be a game changer in turning the pandemic around. Incidentally, and this is a slight digression, lenacapavir itself works by targeting the HIV-1 capsid, and in so doing blocks the import of the HIV-1 capsid into the nucleus of infected cells. It turns out that we actually have a host protein that does a rather similar thing. It's another antiviral protein that we discovered, MX2. It's a rather recently evolved activity. It seems to be present really only in primates and closely related species. MX2 is related to a broader set of so-called MX proteins, but it's sprouted an N-terminal extension that has a nuclear localization signal that actually causes it to dock at the nuclear pores of cells, and in so doing, it inhibits HIV infection and infection by a number of other viruses, it turns out. So lenacapavir, in a sense, mimics what nature came up with previously in terms of an antiviral strategy. So in the last part of the talk, I want to talk about a completely different kind of host defense that was discovered using HIV-1, but may have applications outside the field of HIV. This was not intended to be a project on host defense mechanism, but it just turned out that way. In fact, what we were trying to do in this first set of experiments was to understand why HIV has a rather unusual nucleotide composition in its RNA genome, and whether any of these structures that are predicted to form in the approximately 10 kilobase HIV RNA, whether they have any function over and above what was already known about TAR elements, rev response elements, packaging sequences, and so on. And so what we did was to make a set of viruses, A through P, where we took blocks of the HIV genome and we mutated them in such a way as to change the nucleotide sequence, but not to change the protein sequence. So these are synonymous or silent, otherwise silent mutations. Okay? So different RNA sequence, same protein sequence. We found lots of interesting things using this approach. It's a type of experiment that grant reviewers hate because it's open-ended and not driven by any particular hypothesis, but it's been an extremely rich experiment for my lab, and it's one I would encourage you to do, if not write a grant about. So I'll focus on one particular mutant L. It's not the only mutant that has this phenotype, but it's informative here. So there are 145 synonymous substitutions in this roughly 450 nucleotide block. Those synonymous mutations together are lethal to HIV replication. Wild-type virus grows fine, the L mutant doesn't grow at all. However, we couldn't map the mutations that were responsible for this replication defect, because when we made 4 derivative mutations that cumulatively have all of these substitutions, but individually have a subset, they all grew fine. So clearly, it's a cumulative effect. Now, where the story broke was that if we divided the mutations not into numbers, into segments, but into particular types, what we found was that particular dinucleotides, C followed by G, was uniquely lethal to HIV replication. So just to digress for a moment, CpG dinucleotides are very rare in the genomes of many vertebrates. That's because we have enzymes that recognize CpG dinucleotides in DNA and methylate them. Those methylated nucleotides at a rare but non-zero rate are spontaneously deaminated through a process of oxidation. That causes a methyl C to T change. Absent the methyl, you get a C to U change. U is recognized as foreign, repaired, and the correct base opposite the G nucleotide put back. But when you get a C to T change, DNA repair is confused about what's the correct base. And so over hundreds of millions of years, in vertebrates, CpG dinucleotides have been gradually purged from our genome, so we have about 1/5 the expected number. That's a well-known, well-documented phenomenon in DNA-containing organisms such as ourselves. What's strange is that the RNA viruses that can't access that mechanism, but infect those hosts, mimic that property, that loss of CpG dinucleotides. And HIV is no exception. Very few CpG dinucleotides. But what we found is, if we took this L mutant and put the 37 new CG substitutions that we had put in that original L mutant, that was lethal. Okay? The other mutations, 108 of them, no problem at all. Something unique about CpG, because if we just switch the order and put as many GC dinucleotides, no effect. Virus replicates fine. It's something special about CpG. So at this point, we thought, well, maybe there's a host protein that somehow recognizes CpGs and causes that replication defect. So we did a screen, infect cells with wild-type or CpG-enriched virus, put an siRNA focused library in and measured infectious virus yield, looking for genes that would specifically, whose knockdown would specifically rescue that replication defect. This is the hit that we got. It's called zinc finger antiviral protein. It had previously been identified as an antiviral protein, and its overexpression had been shown to inhibit replication of several RNA viruses by Steve Goff and others. But it was completely unclear how ZAP works, how RNA is recognized. When people had tried to map target sites for this protein, they basically slipped through their fingers in ways that were kind of reminiscent of what happened to us when we tried to map what was responsible for that L mutant phenotype. But here's the key experiment, though. If we take wild-type virus, it replicates in cells that have or don't have ZAP or have been reconstituted equivalently. But if we take that virus that has about 37 CG mutations that's dead, if we knock out ZAP, now it replicates just like wild-type virus. Clearly, ZAP's necessary and sufficient for this phenotype. With Janet Smith and her team at University of Michigan, we've solved the structure of the N-terminal domain of ZAP, which is the business end of the protein, in complex with an RNA, and it becomes obvious at the atomic level how this protein is specifically recognizing RNAs that have CG dinucleotides in it. This is the crystal structure, and it has two pockets that can accommodate a C and a G, and only a C and a G nucleotide in the midst of this RNA binding cleft. Work from others, particularly Chad Swanstrom and Stuart Neal, we've contributed to this a bit, has shown that basically how ZAP is working is, in addition to recognizing RNA, which we've shown, it recruits a nuclease, and in so doing causes cytoplasmic depletion of RNAs that have too many CG dinucleotides. So I told you that this has happened in vertebrates. So ZAP is present in vertebrates. It has its origins around the time tetrapods evolved. So everything sort of downstream of here has a ZAP protein, but everything that diverged prior to that does not have a ZAP protein. So ZAP evolved after a major split, functional split between invertebrates and vertebrates, which is essentially the acquisition of this CpG methylating phenomenon. So ZAP only occurs in organisms that have the ability or have evolved CpG-poor genomes and have CpG-poor viruses. It's not present in insects and arthropods, which generally speaking have viral genomes that mimic the nucleotide composition of those hosts and are CpG-rich. They have just the expected number of CpGs based on random base frequencies. But we think that the fact that these things have been doing this hundreds of millions of times per day for hundreds of millions of years has put an evolutionary pressure on vertebrates to essentially defend themselves against these viruses to which they are exposed constantly. And so this gives, we think, the explanation for how and why ZAP has evolved. It also gives us what we thought was an opportunity. That is to try and rationally attenuate viruses by recoding their genomes, by putting back those missing CPGs, essentially driving the virus into the teeth of ZAP as a means of rational attenuation. And I'll tell you about that work in the last few minutes. So we tried to do this sort of initially just by adding lots of CPGs into the viral genome. And we found, yes, we could get viruses that didn't grow very well, but sometimes that was ZAP-specific and sometimes not. So we undertook a rational series of experiments to really define what is an optimal ZAP target. And so we varied number of CPG dinucleotides in the genome, the spacing between them, and surrounding nucleotide composition. I'll just show you one experiment. So we took HIV-1, took that L segment that I told you about, and added 1 to 23 CPG dinucleotides in that segment. And you can see the growth curves here. Let's look at this graph here. These viruses all grow equivalently in ZAP knockout cells, but adding one CPG has a statistically insignificant effect on virus replication, but as you add more and more, you can accrue 100-fold replication defect. We did the same experiments with spacing and surrounding mononucleotide composition, and what we found was that about 15 CPGs are sufficient to completely attenuate HIV-1. Spacing is optimally 12 to 30 nucleotides, which makes sense when you know how big ZAP is. And that if you make the surrounding genome as single-stranded as possible, make it A-rich as possible, HIV is already pretty A-rich, that also increased ZAP sensitivity. So we thought we had figured out the rules, as it were, of how to make a virus ZAP sensitive. How do we test whether we have, in fact, deciphered those rules? Let's go to a completely different virus system, a virus that has no homology, a completely different replication style. Can we engineer that virus to be ZAP sensitive? And incidentally, make a vaccine, a potential vaccine for this virus, which causes hand, foot, and mouth disease. It's the scourge of daycare centers around the world. Those of you with young kids will have heard of this disease. We fear it not really because of the disease itself, but because rarely this virus can cause very much more serious outcomes, including polio-like illnesses, long-term cognitive impairments, and up to and including death, and has done so in some outbreaks, particularly in Asia. So we took this virus, took a segment of this virus. Here's the wild-type virus grows equivalently in ZAP plus and ZAP minus cells. If we make that segment of the genome as A-rich as possible, make it as single-stranded as possible, we get a subtle ZAP phenotype. If we put a lot of CG dinucleotides, we get a subtle ZAP phenotype. But if we do both, we get a virus that is not completely, attenuated to a large extent by ZAP. Now, we also made a mouse model in interferon receptor knockout mice. We infected them with this mouse-adapted version of this virus, and they get a polio-like illness with limb paralysis, and they have to be euthanized within the first couple of weeks after infection. We infect them as neonates. And so that's documented here. Here's the mouse-adapted virus, wild type. Here's a clinical score and a Kaplan-Meier curve. We've made ZAP knockout mice, but that doesn't matter for the wild-type virus because the wild-type virus isn't susceptible to ZAP. But if we take the virus that I just told you about, that we engineered to be attenuated in a ZAP-dependent manner, it's indeed attenuated in ZAP-deficient mice, but continues to be lethal in ZAP knockout mice. And that's completely an effect of a virological effect of virus replication. You can see the amount of virus is determined by CPG dinucleotides and ZAP. And so if we take this virus, okay, this attenuated virus, okay, and we infect wild-type female mice, okay, They shrug it off. They do well, okay, because the virus is attenuated. But what they also do is make high-titer neutralizing antibodies to that virus, okay, which is shown here. Those antibodies block infection. If we allow those female mice to grow up, mate them to naive males, and then take their offspring, which acquire antibody from their mothers through milk, we challenge them with the virulent wild-type virus. Now, if their mothers had not received the attenuated virus, they get paralysis and die. If their mothers had received the attenuated virus, made antibodies which they transferred, they are largely protected. This is a couple of mice that actually died for other reasons. First-time mouse mothers are not the best mothers, so it turns out. These actually didn't die of the virus, but we have to record them as deaths anyway. But what I hope this shows you is that by understanding how ZAP works, by programming a virus to be sensitive to this innate viral defense, we can rationally design an attenuated virus that can, in principle, work well as a viral vaccine. And in principle, this approach could work for essentially any virus or any RNA virus. Okay, so that brings me to the end. Let me summarize. What I hope I've shown you is that there are a wide variety of host defense mechanisms among animal species that can strongly limit their host range. The raison d'être for HIV accessory proteins, and I'm sorry, that's the only French I'm going to speak today, is to antagonize these host proteins. That's their major and perhaps their only function. In devising antiviral strategies, I alluded to the gain-of-function experiments that nature does every day. It has been far more inventive than humans in coming up with ways to inhibit viruses. But we can learn by making drugs like lenacapavir from nature to make new antiviral strategies. And perhaps most importantly, engineering resistance to antiviral proteins is a key way for engineering viral tropism and generating animal models. And engineering susceptibility to antiviral proteins, I hope I've shown you, is a potential way to generate live attenuated vaccines. Okay, let me finish by acknowledging people who have contributed to this work. Most importantly, Theodora Hadsioanu, who's my wife and scientific partner. We run our lab together. The work on the simian tropic HIV-1 is as much hers as it is mine. As I mentioned, we've worked closely with a group at NCI Frederick, including Jeff Lifson, Vineet Kaur-Romani, who was instrumental in starting this project. We've worked with them on simian tropic HIV-1. Work on antiviral proteins has involved a long list of people, but perhaps most notably Stuart Neal, who discovered tethering, David Perez Caballero, who figured out how it works, Shalini and Sam and Melissa Kane particularly for the discovery of MX2 as an antiviral protein. Matthew Takata, he's responsible for initiating the work on ZAP, discovering its CPG specificity. And Daniel Gonçalves Carneiro did all the work to derive the optimized vaccine virus. And the structural biology I showed you was in collaboration with Janet Smith and her team at University of Michigan. Thank you very much for listening.
Do you have time for a couple of questions? Eric, are you going to the microphone? Or all right. So Eric will ask.
Very nice talk, Paul. So with ZAP, how does it tie? Does it tie in to like the MyD88 signaling cascade, like CPG DNA? And what's the downstream effect of ZAP?
ZAP appears to have really no effect, no ability to transduce a signal. So it's clearly recognizing cytosolic RNA that was made in that cell, not RNA that was internalized in an endosome in the way that the TLRs would. We've made ZAP knockout, ZAP competent cells, put RNA on them. ZAP itself does not seem to change gene expression in the way that you hypothesize it might. What it does do, and we have a paper we're just working on at the moment, it has subtle effects on the small set of human genes that they themselves have a little more than average number of CPG dinucleotides. So there, in that sense, it's probably acting directly on those RNAs rather than acting as a sensor.
But as you change the sequence, you probably are moving towards like a RIG-I approach, RIG-I signaling as well, or not, in the ENV, like getting closer to induction of that cascade, or is it solely ZAP that you see?
So we do not see any signaling effects of our mutant viruses, at least in the cell lines that we have worked in, or in PBMCs.
Hi, my name is Ryan. I don't know if Dr. Arts might have just asked the question I'm about to ask, so let me know. But I noticed your CPG mutations were only in the envelope section.
Yeah.
My question was, have you tried it in the other parts of that HIV genome, and like, what happens? Does location matter for this mutation?
Not over and above the effect of a local nucleotide context. So we have done experiments where we put CPGs in spot A and spot B and seen different effects. But then when we look closely, we see, that spot A was a little less A-rich than spot B, and when we normalize for those differences, we get basically the same outcome. It's not a specific property of ENV by any means. You can do the same thing in POL, for example.
Okay, thank you.
Hi, Jerome from
Very
Kelix.
quick questions, just.
Quick,
Yeah.
difficult. Anyway, for the experiment using the non-mutant primate in pigtail, you are using the modification in terms of the VIF using the human VIF. But if we are looking about the kinetics of the virus replication in these viruses, is spontaneous control. If you delete with a CD8, you increase the viral load. So do you think that we are replicating a little bit what we found with HIV delta NF, like? Because after the challenge is protecting. So it seems to be like a delta NF viruses. Are you agree or not?
Clearly, those monkeys have an elite control type phenotype, right? And if you deplete CD8s, you get a blip, and then you get control. Clearly, the immune system is on top. I don't think that is necessarily the same as you'll have to refresh my memory what happens if you give a delta NF virus to a CD8 depleted macaque.
So thereafter you have a deletion and progression, and you do a second infection with a delta NF, you have a long protection.
Right, but the initial infection with delta NF in a CD8 depleted
You
macaque,
increase.
they go to AIDS?
Yeah, not
Okay.
go to AIDS, but you increase, in particular in a juvenile
So
animal.
so it's sort of halfway between what we have here then. Yeah.
All right, we will just hand Paul a gift and thank him once again for this great presentation.
Thank you.